Hello everyone, I'm Shaoheng from MD Anderson Cancer Center. Today I'm going to talk about our work on latent periodic process inference from single-cell RNA-seq data. It's important to study the changes of cells among development, differentiation, and pathogenesis. Ideally, we would like to track a single cell over time to see how the gene expressions change. However, in reality, although single-cell RNA-seq is of very high resolution, the cell is destroyed, was measured, and cannot be profiled over time. The expression data we have is a profile of many different cells. Nevertheless, we can build a trajectory from those different cells and assign a pseudotime for each cell with regard to its location on the trajectory. That said, pseudotime is different from time, and trajectories may end at where it starts, and that's what we call a circular trajectory. A prominent example is the cell cycle. A cell starts at G1 phase, in which it ensures everything is ready for DNA synthesis, and then in the S phase, the DNA will duplicate, and the cell goes into the G2 phase to ensure that everything is ready for the final dividing, and actually divides into two in the M phase. After that, the two daughter cells will be very much like the original cell in the G1 phase. As a matter of fact, many genes directly or indirectly related to the cell cycle will go up and down in different phases. That's very important information for us to recover the trajectory. Besides cell cycle, there are also other circular trajectories, like the circadian rhyme and the onset of malaria. There are also other processes that can go back and forth and potentially form a circular trajectory. For example, the Epithelial-Mesenchymal transition, EMT, and its opposite, MET. Here, we assume that a circular trajectory is controlled by sets of genes that are turned on and off at relatively precise timings, and use a method called cyclone, which is based on autoencoder, to discover the circular trajectory, measure the circular magnitudes of different genes, and remove circular patterns to reveal other information of interest. Autoencoder is a well-known method to compress high-dimensional information into low-dimensional embeddings. It forces expression data to be encoded so that it can go through a bottleneck. It optimizes the encoder and decoder so that the reconstructed expression is as similar to the original expression as possible. Although the embeddings include rich information, it is often hard to interpret. It would be great if we can have one component in the embedding that represents the circular trajectory and other components for other changes in the cells. So how can we make this happen? Our solution is to use periodic functions, sine and cosine, as the activation functions instead of the widely used ReLU or sigmoid functions for neural networks. It's like doing a Fourier transform on time series data, where the time information is erased. Because sine and cosine have a period of 2 pi, there will be no difference for embedding x and x plus 2 pi. So we can just take x mod 2 pi as the embedding, drawing it on a circle to represent the circular trajectory. So how does this strategy work? We tested cyclone on a mouse embryonic stem cell dataset, including 288 cells and more than 30,000 genes. All cells are actively proliferating. The cell cycle phases are labeled by flow cytometry and discretized into G0, G1, S, and G2M stages. Each stage has 96 cells. We tested if cyclone can discover the circular cell cycle trajectory. Each line in the inner circle corresponds to a cell and the outer circle shows the distribution of cell from each cell cycle phase. The colors correspond to the stages: red for G0, G1, green for S, and blue for G2M. Those are labeled using the ground truth. Cyclone is a tool to infer the circular trajectory, but it doesn't have the ability to discretize the trajectory into the phases, because it is fully unsupervised and does not know the markers for each phase. What we can see is that the three phases are well separated over the inferred pseudotime. We can also inspect the gene expression over the pseudotime using a heat map. Each column of the heat map corresponds to a cell. The cells are ordered by the pseudotime. You can imagine that the left edge of the heat map can be wrapped around and attached to the right edge. It's apparent that there are many genes that have a periodic pattern over the pseudotime, and many of them are not traditionally defined cell cycle markers. We will show that utilizing the additional information from those genes may help fight the noise in the cell cycle marker genes. Compared with the two previously showed images, the inferred cell cycle trajectory is very similar to the illustration of cell cycle, and the heat map also corresponds to the periodic changes of molecules in a cell. We compared with other methods how well the cell cycle phases are separated on the pseudotime. We find that cyclone works better than the methods using marker genes or supervised model, including slalom, cyclone, and recat. Being able to consider periodic pattern in all genes may be one of the reasons. It also works better than OSCOPE and Cyclops, which also target circular patterns. It outperforms a few other decomposition-based methods, including non-negative matrix decomposition, independent component analysis, and principal component analysis. To show that cyclone is robust to variances among datasets and technologies, we performed the same comparison on a few other datasets that are generated by qPCR, including a prostate cancer cell line PC3, metastatic breast cancer cell line MB, and a lymphoma cell line H9. Cyclone outperforms all other methods on all datasets. Cyclone also scales to larger datasets. We tested it on a human embryonic stem cell dataset with more than 10,000 cells. Because there is no ground truth label, we used GSEA enrichment score of cell cycle gene set as a criterion. It shows that cyclone returns the best result within a reasonable period of time. We also tested if the number of cells or number of genes affect the accuracy of the methods. We subsampled the cells and genes and tested cyclone against recat. The left panel is for subsampling cells. The number of cells is for each stage, so the total number of cells is 3 times as large. Lower numbers of cells introduce more variances into the result, but the average performance generally holds. Number of genes in the red panel is more interesting. Because recat is based on marker genes, when the information of those genes are lost because of the subsampling, the performance drops fast. Cyclone, on the other hand, considers the circular pattern in all genes and can reliably recover the trajectory from as low as 2,000 genes. It is also a common need to remove the cell cycle from the expression profile because the cell cycle may overshadow other processes. For example, the cell cycle may confound the detection of the subclones in a tumor, and detecting of such subclones may be vital for a treatment. To test that, we used the MESC dataset to simulate virtual subclones. We randomly chose cells and genes in the dataset and doubled their expression to create the new subclone. We used the cyclone to discover the cell cycle pattern and remove it from the profile. We tested how separated the subclones are after the cell cycle is removed. Here, we show an intuitive comparison of the separability of subclones before and after the removal of cell cycle patterns. The colors correspond to subclones, and the shades correspond to cell cycle phases. Before the cell cycle is removed, the subclones are mixed because the variance is dominated by the cell cycle phases. After its removal by cyclone, the subclones become the most prominent factor in the single-cell profile. Indeed, the separability increased from around 0.7 to around 0.9. We also compared it with the regress out in Suret and CC remover. Both methods use marker genes. We also removed each of the principal components as a baseline. It turns out that cyclone performs significantly better than all other methods. One reason is that the cell cycle markers may also be perturbed in the subclones, which makes it necessary to infer the cell cycle from all available genes. Next, we use cyclone as a metric for the periodicity of genes. We use the HESC dataset, which compares a nicotine-treated sample with a control sample as an example. We run cyclone on each sample to capture the cell cycle trajectory and use the magnitude of the circular component on each gene as a measurement of the periodicity. We scale the genes before the procedure so that the metric is standardized. We'd like to find the genes whose periodicity changes significantly. In the figure, each point represents a gene. The x-axis is the periodicity in the control group, and the y-axis is that in the nicotine-treated group. It can be observed that the periodicities of most genes don't change a lot between the two groups. It verifies that this is a consistent metric. There are also a few conspicuous exceptions that are far from the diagonal. For example, the LDHA and the ENO1, which are more periodic in the nicotine-treated group. Indeed, those are genes that are known targets of nicotine. They play roles in cell proliferation and are also related to lung cancers. There are also a group of mitochondrial genes whose periodicities are lowered. They also appear to be targets of nicotine suggested by other studies. The metric can suggest more potential genes of interest, other than those that are up or down regulated. The transitions between epithelial cells and mesenchymal cells is vital in organogenesis, and is also hijacked by cancer to cause metastasis. It's natural to ask if EMT and MET form a circular trajectory. To acquire some insights, we found a dataset with around 300 epithelial cells and 300 mesenchymal cells, where there is known to be EMT happening. We run cyclone to capture a circular trajectory and test if the circular trajectory corresponds to MET and EMT. Again, we use the circular plot to show the embeddings. The red lines are for epithelial cells, and the blue lines are for mesenchymal cells. We find that the two types of cells are well separated by cyclone. But are there two types of transitions? We picked out the cells near the two transition points and found differentially expressed genes between them. In the results, we find a few unique MET marker genes which don't show up in the EMT gene set. This means that the two transition points may really respond to EMT and MET respectively. To sum up, cyclone can be used to reliably embed the cell cycle, remove it from the profile, or discover the genes whose periodicities change significantly. Cyclone can also be used to analyze other circular trajectories like EMT and MET. Cyclone is efficient and robust. It also scales better to larger datasets. There are a few possible extensions. We've already tested that it's possible to inject prior knowledge into the model by giving marker genes higher weights. The performance usually increases a little bit, but there is really no free lunch, and the result always depends on the scenario. There are also a lot of single-cell data analysis methods based on neural network published in the past few years. The core of Cyclone, the periodic unit, can be integrated into those tools easily and to make new findings. This work is published in Nature Communications earlier this year. You can have a look for further information. The software is available on GitHub. I'd like to thank my advisor, Dr. Ken Chen, my co-authors, Dr. Fang Wang and Jincheng Han, and all other group members.